Okay, so this is the opening picture that actually got in the design section got suggested, and I wish that I had seen it for one of our earlier chapters, because you can see there's a Kirby-Bauer test back here, as well as some selective and differential media, but whatever. Even though we're past this, I couldn't resist using it for the first slide, for the title slide. So chapter 14 and 15 go together, and they are focusing on the host defenses. So chapter 13 was all about how the microbacteria, the bacteria and viruses, and how all the organisms can attack the human host. And chapters 14 and 15 are going to be about how the host responds, okay? Which we did not consider very much in the beginning when we just talked about chapter 13. So this first chapter here, 14, is all about the innate or non-specific response. And these two blue boxes, the first line of defense and second line of defense, that's what we're going to go over today. Okay, all of these are included in chapter 14. This other side, this other arm of our defense, the acquired response or the specific response, the immune response, that's the next chapter, that's chapter 15. So we're not going to talk very much about that today. We're going to focus on the innate or non-specific defense today. We're going to start with simple stuff, physical barriers, and what they mean by physical barriers in your book, they mean mechanical barriers. Barriers that specifically prevent bacteria, viruses from being able to either enter or from being able to adhere. Because remember, when we talked about the course of disease, we talked about portals of entry, adherence or adhesion, and then the ability to set up an infection, right? So this slide is a good summary of all of those, both the physical and chemical barriers. And on your note outline, I made a list of some highlights of the physical barriers. And I would remind you that places where there are physical or mechanical barriers, usually there are also chemical barriers to go with those mechanical barriers. So for example, if we're looking at the ciliary escalator in your respiratory tract, right? Pretty much concentrated down here right before the split in the bronchi, and that its job is to trap particles, and these cilia constantly beat upwards so that you can expel those particles, right? So that would be an example of physical barrier, because the mucus is moving. In this case, it's moving upward. And it would also be an example of chemical barriers, because there would be lysozyme there in that mucus that would help kill the bacteria. So I'm not going to go through each and every single one of these. You can look at the ones that I picked out on your handout to focus on. I would also remind you that, and I don't think I put this on there, although I meant to, that your normal microbiota are also secreting both bacteria and fungus that live on you permanently. They're secreting technically antibiotics that are going to also contribute to keeping the pH of your skin low, as well as they're going to secrete substances that are going to particularly attack and maybe even kill other fungus and bacteria that are trying to cause an infection. Your textbook divides out the systems involved in the immune defenses into 4 parts. Okay, this is relative to this textbook and the way that you're going to find these questions when I assign in Connect when you go there, which is why I'm doing it this way. I probably wouldn't do it this way normally, but that's not important, right? So the first system that they talk about a little bit here is this mononuclear phagocyte system. And this woman, you see where these purple areas are on her body. That's supposed to be illustrating to us high concentrations of this particular phagocytic cells where they're located. I would point out to you, notice they're located primarily in the trunk, right? Not so much in our appendages, but to protect most of our vital organs. And this system consists of, you can see some macrophages here and neutrophils. These cells, the ones that are yellow with that are much larger, those are supposed to represent cells in the tissue, okay? Whatever particular tissue is being attacked by an infection at that particular moment, okay? The second part of those systems that are involved in immune defenses as your textbook defines them. And what they're trying to communicate here is that the fluid that is in between both the phagocytes, the lymph, and the bloodstream, so what we would call tissue fluid, this is the place where a lot of these cells can communicate to each other, okay? And you can see these arrows indicating that chemicals and compounds, cytokines, are going to be moving into the bloodstream from the tissues, from the lymph, and from the bloodstream out, okay? This panel is to specifically highlight the organs, or organs and tissues, we would say, particularly of the lymphatic system. Again, which we'll talk about how these lymph nodes work a little bit more when we talk about the next chapter, but this is sort of orientation. This gentleman here is to represent the circulatory system. This lymph woman standing next to him. Notice how all of the lymph vessels, lymphatic vessels, they're pictured in green here, and notice how most of them dead end in these lymph nodes. Okay, again, highest concentration of those is going to be in the trunk and here at the base of your head and a little bit around the brain to protect our most vital organs. And then they list out all the various organs here. This panel is to remind you how connected the lymph and circulatory systems are. So the fluid that is being pumped around the lymph, right? It's going through the lymph and traveling and can move back and forth between the lymph and the bloodstream. Eventually that's going to end up being pumped and decreased in volume as it gets pumped through the heart. All right, the specific cells, which we would call the stem cells and the effector cells, are on this panel. And what I want you to don't over study the specific effects and the specific jobs of all of these cells. What I'm interested in that you understand from this particular panel is that all of these cells are derived from stem cells in the bone marrow that we call hematopoietic stem cells, meaning they can become any cell of the blood, either a red blood cell, a platelet, or cells of the lymph. Okay, these are early differentiated cells. See how it splits very early into stem cells for all blood cells except lymphocytes, and then lymphoid stem cells. And then these, of course, are going to differentiate into B cells, T cells, and natural killer cells. The rest of these myoblasts are going to become the granulocytes, the monoblasts for our monocytes are our phagocytes, and then red blood cells here and the megakaryoblast. Most of these cells, I'm going to probably ask this question to you as a multiple choice, which of the following is not correct, and I'm going to list the precursor cell and then the effector cell, and 3 out of the 4 or 4 out of the 5 will be correct and 1 will be incorrect, and your job will be to pick out the one that is incorrect. Okay, now if we could look back to our picture that we started with, right now we can start talking about this whole second line of defense because it involves some of the cells we talked about, some of the tissues and fluids that we talked about, and some pieces of the immune system. So now we're going to march through these 4 areas where we have both cells and the compounds they release, cytokines, lymphokines, I guess mostly cytokines, and chemical factors that are going to affect how well we mount a non-specific or an innate response. Remember, these are the same for all organisms. These are not specific to a particular infection or a particular bacteria or a particular virus. So let's look at phagocytosis first. This is panel 14.5 in your textbook, and your book has divided it out into 7 steps here. I'd like us to just take a look at some of the ones that might be new to us or steps that aren't overemphasized in other places that you've learned about phagocytosis. This is to represent the whole cell, this picture up at the top here, and notice they say chemotaxis by the phagocyte. So the phagocyte needs to be attracted and sort of needs to migrate toward where these bacteria are. And one signal to help get it there is these proteins and sugars that are on the surface of bacterial cells. They're called PAMPs for short, and that stands for pathogen-associated molecular patterns. Now, what does that mean? Well, it means things like peptidoglycan, something that we as humans don't make. It's foreign, but it's not usually an antigen by itself. So it's a pattern of sort of like a repeating pattern. A lot of these are going to be carbohydrates that are going to be able to attract phagocytes, and the phagocytes, look, they have receptors for them. The phagocytic cells that we're mostly talking about here, these are going to be monocytes that once they engulf a bacteria and degrade it are going to mature into a macrophage, and neutrophils. Those are probably the two best cells that have the most highest phagocytic activity. Stepwise, the bacteria are engulfed by those receptors. They're put into a vesicle. That vesicle is called a phagosome, and it has particular markers on it that are going to attract lysosomes from inside the cell to merge with that phagosome. When those lysosomes merge and start releasing their enzymes, it's referred to as a phagolysosome. And then, of course, the bacteria is going to be destroyed, broken down into pieces. Notice this list of enzymes here. Those would all be coming from the lysosomes, right? And then the release of residual products. So these are things the cell can't use, as well as a subcompartment of this when the killing and destruction takes place. This is the timing point where if this is an antigen presenting cell or an antigen processing cell for the immune system, we would see the joining up of some class 2 MHC and putting that out on the surface as a marker for B cells or T cells. Inflammation, that's sort of the second major set of second line of defense. Your book divides it up into 4 major parts, right? I've listed them on your handout as well. This is figure 1418. They divide it up into early events, vascular reactions, edema and pus, and then the resolution. Just real quickly, go through this with you. Notice the green bacillus here that are being put into this wound. Ouch, I've stepped on a nail, right? Or I've cut myself. In order for inflammation to be triggered, there has to actually be tissue damage. So this can be physical damage, like in this picture, or it can also be chemical damage. So if, for example, you have bacteria that are producing an exotoxin from the last chapter, and that exotoxin has a specific target cell, and it destroys those cells or degrades those cells or kills those cells, that would also be able to trigger an inflammatory response. Notice that these early events, we've got tissue damage, that's going to release some cytokines, as well as these mast cells that are in the area in the tissue. They're also going to release some chemical mediators. And one of the first things that's going to happen is that the vessels nearby are going to constrict or get smaller. That's to help prevent excess blood loss. Once the clot is formed and you're no longer actively bleeding in this area, and you can still see the tissue damage here, this little vessel's been severed, right? Now, if there are bacteria in the wound, that's when the neutrophils and the phagocytes, the macrophages, are going to squeeze out of the blood vessels and get into this tissue and actually start killing and degrading those bacteria. How do they do that? It's a special little process that has a special name. It's called diapedesis, because the cells are able to stick first, that's the margination, and then see how when the vessel is dilated, how there's little gaps between the cells and the endothelium here, so that the cells these macrophages and neutrophils, now they can squeeze out these tiny little holes and move into the tissue. Notice the blue arrows, they're following a chemical gradient, okay? We call the factors that are chemotactic are factors that are going to attract macrophages, neutrophils, right? And that the chemotaxis is the actual movement of those cells from the blood into the tissue. Once they get in there, notice there's some swelling now and there's pus formation because these cells they're doing a lot of work. They're breaking down all those bacteria, they're producing water, they're producing a lot of byproducts. And some of these cells are also dying themselves, which is going to call in other cells to clean up the mess. And that's going to contribute to the swelling, the fluid buildup, and then that pus would be filled with dead cells, dead white blood cells, neutrophils, macrophages, some dead bacteria parts, and fluid mostly. And then the fourth step here, you can see that now the vessel's been repaired and there's a scar formed, the tissue, the skin surface has also been repaired. And notice now you have some other cells entering the area. They've actually labeled them as lymphocytes to finish with the cleanup. And notice how large these cells have gotten. This is supposed to represent a macrophage, and that would be a monocyte that's been ingesting these bacteria and was busy working in step 3 there. Your book mentions that fever is often accompanies inflammation. And I just put these 2 slides in so that I didn't have to spend a lot of time talking about it. I also listed some of this on the handout that I gave you. What's a little bit more interest to us are the pyrogens, the compounds that can actually change the settings in the hypothalamus so that we actually experience a fever. And those are going to be divided into, of course, things that come from outside the body, exogenous pyrogens, and endogenous. These, I think, we think of pretty easily, right? So these are going to be triggered by the bacteria, virus, fungus themselves. I listed specifically here endotoxin, the lipid A portion of LPS. Again, back to chapter 13, and also if you think all the way back to chapter 4 when we talked about the various compounds that are in the bacterial cell wall. Endogenous, or originating from inside the body, some of these cells that were just called in for inflammation, some of the cytokines that they produce, some of the the factors they produce can be a pyrogen, and they can instigate a resetting of the hypothalamus, particularly a couple listed here, interleukin-1 and tumor necrosis factor. Benefits of fever, again, I put them in your handout as well. Some organisms are very temperature sensitive, and so even a slight change in the host temperature will actually kill or inhibit these organisms. Most bacteria scavenge iron from our bloodstream, and it's necessary for their nutrition. And when you have a fever, that iron is reabsorbed by our bodies, which makes it more difficult for the bacteria to get the iron they need. And last but not least, having a fever actually does increase your metabolism. So think about production of ATP, think about mitosis, cell division, being able to produce more proteins. Think about more protein synthesis, which requires a lot of ATP. If we're going to be making immunoglobulins, if we're going to be making all of these chemical factors that are being released, most of them are proteins. So we need the ATP in order to make all those proteins so they can help us out. Now I'm going to flash back to this first picture again, phagocytosis, inflammation, fever, and here we are at the last one, antimicrobial proteins. And your textbook goes through the description of 3 sets of antimicrobial proteins, and I would say by far the most complex of those is complement. So I'm going to spend the most time now talking to you about complement and how it gets activated, particularly the classical pathway and the alternative pathway. And then we'll spend a little time on interferons and the antimicrobial peptides. So for complement, I am going to switch, if I can here, to a whiteboard, and I'm going to try to draw a little bit for you some of the major steps of the complement cascade. Okay, so complement consists of 20 or more proteins that are always in your bloodstream, zooming around, and that most of them are in an inactive form until they are triggered to find each other, bind together, become active. So here I'm going to draw for us a bacteria. Of course, it's going to be, it looks like our friend E. coli. And on the surface of that bacteria, to activate the classical pathway, what you need are immunoglobulins. There we go. So I'm going to draw some immunoglobulins, and it's usually, quite honestly, it's usually IgG. So those of you that already know a little bit about the immune system, you might recognize that crude drawing there is the immunoglobulin. And once that occurs, that's going to facilitate, let's make it pink. This protein, it's called C1 for complement protein 1. C1. And C1 is going to bind to those immunoglobulins. And that is then going to attract some other proteins. Let's see. I'll make them green. It's going to attract C2 and C4. Okay. I'm just going to write the numbers, not the letters, because that's going to take me so long. So that's C4, this is C2. Okay. When all of these get together, when all three of these, C1, the immunoglobulin, bacterial cell wall, C2 and C4, when all of those get together, what that does is it's C1 is going to be activated now, and it is going to cut the C2 and the C4 into each of them into almost into half. And we call the halves C2A and C2B and C4A and C4B. Put a little arrow here. It's going to break it into two parts. And I'm just going to kind of draw them as a half circle like that. Two little pieces. And the same for C4. It's going to get broken into two pieces. C4A and C4B. Now, the B portions, C2B and C4B, come together to form a new molecule, and it's actually called 2B4B. And if you think about it for just a second, right, this is important in our blood because we don't want complement to be activated all the time, because what we're going to see is the effects of it are really damaging, and they can, if it's allowed to persist, they can affect some of our cells as well. It's actually going to cause the bacterial cell to be lysed. It's going to cause more inflammation and more phagocytosis. Now, this 2B4B that I've drawn here, this is going to attract C3 put that inside. Oh, no, I can't. This is C3, and C3 is then going to be split into two pieces. See the pattern here? And and of course they are going to be called C3A and C3B. It's C3B that we want to focus on. This is the last of these molecules to become enzymatic and be able to have an effect on and split or break apart another molecule, and that's going to be C5. Circle here. It's a little easier. C5. And C5 is also then going to be split into two pieces, 5A and 5B. Let me go back to my line. So this is C5B here. Now, it's 5B, C6, C7, and C8. I'm going to redraw our bacteria friend here and put some of those molecules on its surface. So literally, they form these little tiny circles, and they form this little mound. So now we have got a C5, C5B, C6, C7, and C8 is the last one I'm trying to put on here, like that. And that set of molecules around them, then, kind of looking for a different color here. Let's make it yellow. C9, several C9s go around the outside of that, and then it's almost like a drain plug. C9, C9, C9. And it actually, if you look at it with a microscope, it actually kind of looks like a flower. And the center of the flower would be all of those proteins, the 5B, the C6, the C7, C8. And then it's surround the petals, if you want to think about the petals, are all C9. Let's see, whoops, baby line. C9s. And then what happens is this center plug gets pulled out, and then there's a hole in the surface of the bacteria, and it dies because they're quite large. There's going to multiples of these are going to form all around the bacteria, and then the contents are going to leak out, and the cell's basically going to be dead. Now, the other important piece of this has to do with our friends here, our C3A and our C5A, because those two, they contribute to nonspecific defenses in another way. They increase inflammation. They do that by, there's binding sites for them on mast cells and basophils, and then that's going to trigger inflammation, which of course is going to trigger more phagocytosis and making chemotactic gradients to call more cells to the area. And then lastly, I'll give myself one more bacteria over here. Lastly, I was hoping for him to be purple. Some of the leftover C3b, once it's no longer active, it can actually stick to the surface of the bacteria. And this we're going to see is going to increase phagocytosis by macrophages. So I'm going to kind of draw a macrophage here, right? It's supposed to be the nucleus. I'll just label it macrophage. This is a shorthand for macrophage. Letter O with a line through it. So this would be a macrophage. So this is the increase in phagocytosis as the third benefit to complement activation. Now, this pathway that I've just shown you, that's called the classical pathway. of complement activation. There is also an alternative pathway. And so this one's called classical pathway because it was the first one that people discovered and worked out all the steps. But notice that it requires immunoglobulin. And you only get immunoglobulin to bind to bacteria if you've already had an immune response, a specific immune response to this particular bacteria. So we're talking about complement in terms of it being a non-specific response. And the pathway for that is something we call the alternative pathway. And I'll just use maybe a darker green or maybe this bright blue. And the alternative pathway, what is required is same as before. You do need to have a bacterial cell wall or maybe a fungal cell wall or some parasitic cell membrane. But instead of having these immunoglobulins like we had over here, instead of that, you would require 3 factors. B as in boy, D as in dog, and P as in Peter. Okay, so you need bacterial cell wall, those 3 factors. And technically, you also need a little bit of old C3b from the last time complement was activated that is no longer breaking apart C5. Okay. And then when you have all of those things, or if you have all of those things floating around in the bloodstream, that can take you directly, so all of this directly goes to C3b and activating C3b to break into A and B. And then once that happens, it's all the same. All the same. You get the same protein break aparts and you get the same effect of activating complement. You get C5b, C5a, C5b, 5b, 6, 7, 8, all those C9s to lyse the cell, 5a, 3a to increase inflammation, and toward the end, leftover C3b to help with, this is called opsonization. These proteins, I forgot to tell you, C5b, 6, 7, 8, and 9, they have another name as well. They're called the membrane attack complex, or the MAC, right, for membrane attack complex. There is a third way to activate complement. It's called the lectin-mannose pathway. And the difference for that particular pathway is you're going to also eliminate the immunoglobulin and the C1 binding to the bacterial cell wall. But rather than just taking you directly to C3, that pathway, that one blue too, that pathway is going to take you directly to C2 and C4. So you would need C2, C4. They're going to bind to the host cells. Your cells would have lectin receptors on them, and those receptors are going to bind to a particular sugar on the bacteria or fungus. It's called mannan. And then when you have the sugar plus the receptor, let's make the receptor an R. You have the sugar plus the receptor and C2 and C4. Again, that's going to take you directly to here. 2B, 4B, onto C3, and then it's all the same. All right, questions about that before I go on? Because I think if I stop sharing this screen, when we come back, it'll be gone. It'll just be blank, and I won't want to redraw all of it again. Yes, no, not for now. All right, well, chew on that, and then maybe we can talk more about that on Thursday. All right, so let me go back to this. So this was the slide, doesn't have the picture in it, but this is the slide that will help you with the different pathways, what turns them on, and then what happens, okay? Remember, the outcomes are all the same. Here they've given you the full names of some of those factors. Now, so this, notice, bacteria, fungus, some viruses, and parasites, especially this alternative pathway, slower, less efficient because we're not using antibodies, we're not using immunoglobulin, but these two, the lectin and the alternative, are both truly non-specific. Okay, let's take a quick look here at interferon. This is a protein that your body makes, your cells make in response only to viruses. Predominantly, it's not virus specific, but what they mean by that is every virus that you get, you're going to produce interferon. Okay, interferon, there's 3 different subcategories. There's interferon alpha, beta, and gamma interferon. Depending on the virus, depending on which cells are turned on to produce the interferon, you might get 1 or more of those types at a time, but it's made in very small amounts. And the interferon doesn't help the cell that's infected with the virus. So see our virus particles. Again, think back to chapter 6. Right, so here comes the virus, it's coming in, taking its coat off, right? And so the nucleic acid is there in the cytoplasm, maybe headed to the nucleus. This is going to cause the nucleus to synthesize these interferon proteins, and they're going to be released, and they're going to give warning to the neighboring cells. So the neighboring cells will probably still get infected with virus. See, here it comes from cell 1 to cell 2. But when the virus gets there, these cells have had a chance to prepare, and they produce a whole bunch of proteins that we lump together. We just call them antiviral proteins. And they can actually inhibit the virus from replicating inside the neighboring cells. Doesn't help our first cell. Cell 1 is sort of like a sacrifice, but cell 1 produces enough interferon to help lots of the neighboring cells. So again, I would remind you, if we take a look back to this picture, if one of these cells has been infected with a virus, think about how quickly it could spread interferon, right, to all of these neighboring cells, either through the bloodstream directly or through the reticular fluid here, right, the tissue fluid. And then last, the 3rd type of antimicrobial protein are antimicrobial peptides. These again are made by the host. I've put their key characteristics out here. And one of their key features is they're very highly positively charged. And notice this is the bacterial cell membrane, not the cell wall. Membranes, all cells, phospholipid bilayer. Right, so they're going to bind to that negatively charged phosphate head. And what they do basically is poke holes in the bacterial cell membrane, and then the contents of the bacteria leak out.